Hi, today we are going to learn about stages of tooth development. Different stages of tooth development: initiation stage, bud stage, cap stage, early bell stage, and advanced bell stage. Morphological changes and physiological changes. Morphological changes are dental lamina in the initiation stage, and the bud stage and cap stage proliferation, early bell stage histodifferentiation, and advanced bell stage morphodifferentiation, and formation of enamel and dental matrices apposition. Initiation stage: 6th to 7th embryonic life. Formation of tooth can be seen microscopically between the vestibular lamina and dental lamina. Dental lamina connects the developing tooth part to the epithelial layer of the mouth for a significant period of time. Bud stage: enamel organ resembles a small bud shape. The enamel organ consists of peripherally located columnar cells and centrally located polyhedral cells. The surrounding mesenchymal cells proliferate, which results into condensation into two areas. The area of condensation below the enamel organ is dental papilla. The ectomesenchymal condensation surrounds the tooth bud, and dental papilla is the tooth sac. Cap stage: the deep surface of each ectodermal tooth bud becomes invaginated by the mesenchyme called dental papilla. The ectodermal cap stage covering over the papilla is called enamel organ. The peripheral cells are cuboidal over the convexity of the cap called outer enamel epithelium. The cells in the concavity of the cap become tall columnar cells and represents the inner enamel epithelium. Stellate reticulum: the polygonal cells located in between the outer and the inner enamel epithelium begins to separate due to the water begin to draw into enamel organ from the surrounding dental papilla. The star-shaped cells forms a cellular network called stellate reticulum. The cells in the center of the enamel organ are densely packed and form the enamel knot. This knot projects towards the underlying dental papilla. At the same time, a vertical extension of the enamel knot called enamel cord occurs. The function of the enamel knot and cord act as a reservoir of the dividing cells for the growing enamel organ. The enamel knot act as a signaling centers as many important growth factors are expressed by the cells of the enamel knot, thus play an important role in determining the shape of the tooth. The ectomesenchyme condensation, that is, dental papilla and the dental sac, are pronounced during this stage of the dental development. Early bell stage. In the bell stage, the crown stage is determined. Due to the continued uneven growth of the enamel organ, it acquires a bell shape. It was thought that the shape of the crown is due to the pressure exerted by the growing dental papilla cells on the inner enamel epithelium. This pressure, however, was shown to be opposed equally by the pressure exerted by the fluid present in the stellate reticulum. Outer enamel epithelium. The cells of the outer enamel epithelium flattens to form the low cuboidal cells. It is thrown into folds which are rich in capillary network. This provides the source of nutrition for the enamel organ. These cuboidal cells later assumes a columnar form and produce a dentin. Inner enamel epithelium. Single layer of cells differentiate period to amelogenesis. Tall columnar cells called ameloblast attach each other junctional complex laterally in stratum intermedium by the desmosome. The cells of the inner enamel epithelium exert a strong influence of the underlying mesenchymal cells of the dental papilla, which later differentiate into odontoblast. Stratum intermedium. A few layers of squamous cells form the stratum intermedium between the inner enamel epithelium and stellate reticulum. These cells are closely attached by the desmosomes at the gap junction. This layer seems to be essential to enamel formation. Stellate reticulum. It extends further due to the continued accumulation of intracellular fluids. These star-shaped cells, having a large process anastomosis with those of adjacent cells. As the enamel formation starts, the stellate reticulum collapses to a narrow zone, thereby reducing the distance between the outer and inner enamel epithelium. Dental lamina. Dental lamina is seen to extend lingually and is termed dental lamina, successional dental lamina, as it gives rise to enamel organs of permanent teeth. The enamel organ of the deciduous teeth in the belt stage, the successional lamina, and the permanent successor teeth in the bud stage. Dental sac. Dental sac exhibit a circular arrangement of fibers and resembles a capsule around the enamel organ. The fibers of the dental sac form the periodontal ligament fibers that span between the roots and the bone. The junction between the inner enamel epithelium and odontoblast outline for the dental enamel junction. Advanced bell stage. It is characteristic by the compensation of mineralization and fruit formation. The boundary between the inner enamel epithelium and odontoblast outline the further dentino-enamel junction. The formation dentin occurs first as a layer along the further dento-enamel junction in the region of further cusp and proceeds the pulpally and apically. After first layer of dentin is formed, the amyloblasts lay down the enamel over the dentin in the further incisal and cuspal formation. The enamel formation then proceeds coronally and cervically in the region from the dento-enamel junction towards the surface of the cervical portion of the enamel organ form the Hedwig epithelial root sheath. This hairs outline further the root, thus responsible for their size, shape, length, and number of roots. Thank you.